Okay, so in this lecture, we will talk a little bit more about therapeutic mRNA engineering. So, what we need to understand is every region of the sequence affects performance. How efficiently protein is made, how long it survives, and how strongly the immune system reacts, things like that. So, why even this sequence engineering exists? Because the sequence determines behavior of the molecule. Every nucleotide choice affects protein output, mRNA lifetime, immune response, and the manufacturing yield, for example. So when we design therapeutic mRNA, we are, engineering molecular function. The 5' UTR controls one thing, the coding sequence another, and then the 3' UTR something else. So that's why we engineer each region. Okay, now let's talk about 5' untranslated region. So this region plays a central role in controlling translation initiation. So, which is often the step that limits overall protein production. After binding the 5' cap, ribosomes scans along the RNA until it recognizes the start codon. So that means the ribosome has to physically move through the 5' UTR. So this region must let efficient cap recognition, permit the scanning to proceed, and allow the correct start codon to be recognized. So if those conditions are not met, translation efficiency drops, as you can imagine, and then actually sometimes dramatically. And we just saw that scanning depends on physical accessibility of the RNA molecule. If the RNA forms stable secondary structure, the scanning machinery has to unwind it. Mild structure can be tolerated, but um, highly stable structure slows scanning, and a strong structure near the start codon can severely inhibit initiation. So you can kind of quantify all this, and then we use something called the minimum free energy or MFE, which predicts how thermodynamically stable local folding is. Values near zero indicate relatively open structure, while more negative values indicate more stable folding. Because scanning happens step by step along the RNA, local structure, not just global folding matters. And again, RNA structure does not have the same impact everywhere in the 5' UTR. The region immediately downstream of the cap is particularly sensitive because this is where the initiation machinery first assembles and where scanning begins. So structural barriers, here tend to have a much larger effect than structure further downstream, as you can imagine. So for that reason, therapeutic mRNA design often focuses on keeping roughly, I don't know, first few dozen nucleotides relatively unstructured and avoiding highly stable folding near either the cap or the start codon. And to continue our 5' UTR engineering, one thing is even when scanning of these ribosomes reaches an AUG, initiation may not be automatic. So the ribosome, kind of evaluates the sequence, the context actually around the start codon. A favorable Cossack-like environment increases the probability that initiation occurs at the intended site. Additional AUGs upstream can divert scanning, and upstream open reading frame can reduce translation of the main protein. So effective 5' UTR design not only supports scanning, it ensures that initiation occurs at the correct location. And now let's talk about coding sequence engineering. So here we need to know something called as codon adaptation index, CAI. It reflects how similar a coding sequence is to the codon usage of highly expressed genes in the host system. Common codons are usually decoded faster because matching tRNAs are abundant, while rare codons tend to slow elongation. But what we need to understand is that the translation is not controlled by global averages. It is shaped by local codon patterns. Clusters of rare or common codons, create pauses and accelerations that influence ribosome movement and protein folding. Uh, let's continue our codon sequence engineering. So, a translation is not supposed to be a consistently fast process. Cells actually use local slowdowns to help proteins fold correctly while they are being synthesized. When one domain finishes emerging from the ribosome, let's say, a short pause gives time to fold properly before the next segment appears. So when it comes to engineering, we can intentionally place slower codons, in specific regions. But the thing is, uncontrolled slowing is, different from intentional pausing. Long stretches of rare codons can stall ribosomes and may trigger quality control or reduce yield. So the goal is, controlled pacing, not necessarily maximum speed. Let's talk few more points about coding sequence engineering. Uh, Codon choice is not the only thing controlling speed. The ribosome has to physically unwind RNA structure as it moves. If it encounters a, let's say, a very stable hairpin, it slows down. If the structure is strong enough, maybe it can even stall. In extreme cases, the ribosome may even disengage. So that's why people analyze local minimum free energy across the coding sequence. So we are looking for strong structural barriers that resist unwinding. So in practice, very stable local folds are not that good as you can imagine. Okay, a little bit more on coding sequence engineering. When we design a coding sequence, we are solving, maybe two constraints. One constraint is biochemical, how quickly tRNAs decode codons. The other is physical, whether the ribosome can actually move through the RNA. Either one can slow translation, right? But the most problematic situations are when both happen at the same location. A rare codon inside a strong hairpin is not good. That combination can severely impair translation. So, coding sequence engineering is really about balancing, this decoding speed and structural accessibility across the entire transcript. Now let's talk about 3' UTR. One can imagine 3' UTR is something like a control panel for what happens after translation starts. It influences how long the mRNA survives. When the poly A tail gets shortened, where the mRNA localizes in the cell, and how easily decay machinery can engage. So, 5' UTR controls initiation, and 3' UTR is about controlling lifetime and stability. So, this 3' UTR contains, short sequence patterns that act like docking sites for regulatory proteins. One example is, this AU rich elements, where some motifs can recruit factors that accelerate deadenylation and decay. From an engineering standpoint, if your goal is long-lived expression, you generally want to remove or avoid, these AU-rich elements. On the flip side, people often borrow the 3' UTR elements from naturally long-lived transcripts. Globin derived UTR elements are a common example, because they tend to support stability. I mean, we are obviously simplifying here, but the exact outcome depends on which RNA binding proteins are present in the target cell type you are interested in. And this 3' UTR works as a unit with the poly A tail. PABP binds the poly A tail and helps protect it. And it also supports the translation by stabilizing the mRNA. But the 3' UTR influences when deadenylation starts and how fast it progresses. So certain motifs recruit complexes that shorten the tail sooner, which may lead towards decay. So basically, mRNA lifetime is determined by the combination of the poly A tail design and 3' UTR sequence elements. When we generally talk about um, poly A tail, we think of it as just a protective tail on the end of the RNA. But actually, it's more than that. Proteins called PABPs, they bind along the tail, and that coating helps protect the RNA from degradation. But the same proteins also physically connect the tail back to the 5' cap, creating kind of a circular structure. So this circular structure makes translation more efficient. So here is really important part. This the poly tail is not permanent. Cells gradually shorten it over time, and once it becomes too short, the RNA is rapidly degraded. So it means the poly tail is what determines how long the mRNA stays functional inside the cell. Since we just learned that poly tail controls how long the RNA survives. So its length now becomes a design parameter. A longer tail generally means the RNA will last longer because it takes more time for the cell to trim it down. Uh, Therapeutic mRNAs usually sit somewhere around 80 to 100 120 nucleotides, long enough to be stable. Now there are also manufacturing choices. So you can encode the tail directly in the template, which gives you a uniform length. Or, you can add it enzymatically after transcription, which produces a distribution of poly lengths. So in real design work, you are balancing expression duration with manufacturing practicality. Okay, so now let's talk about uridine content and a bit of a safety engineering. Uridine driven innate sensing can be controlled at two different levels. First is a sequence composition. We can reduce total uridine frequency through codon optimization, and if possible avoid long poly U stretches. Second, nucleotide chemistry. So even if uridine remains in the sequence, replacing it with modified forms like pseudouridine or N1 methyl pseudouridine, I mean during the IVT of course, dramatically reduces immune recognition. So the cap structure is also a major immune checkpoint. Cells use certain proteins to detect RNAs that look foreign, especially those with incomplete capping. Cap 0 RNA is often treated as non-self, and translation gets shut down. So that's why therapeutic mRNA must be properly capped, typically cap 1. Cap 2 exists in nature but is rarely necessary in therapeutics. So a double-stranded RNA is one of the strongest innate immune triggers in IVT mRNA. We usually think of it as a manufacturing impurity, because we remove it through purification process. But one thing we need to know is that the formation is not purely random. A template sequence strongly affects how much double-stranded RNA is generated. So basically, double-stranded RNA control is both upstream and downstream. So we minimize formation through sequence design, and we control residual levels through purification and batch QC. I think historically, this T7 transcription favored G-rich initiation sequences for high yield. But in therapeutic mRNA production, the start sequence is now largely dictated by the capping chemistry. For example, co-transcriptional capping systems such as CleanCap require specific initiation motifs for efficient cap incorporation. So, the first few nucleotides are not really a design variable. They are, defined by manufacturing platform. Okay, so finally, to summarize, what we need to understand is that therapeutic mRNA design is really a region by region problem. In the 5' UTR, the main goal is accessibility. In the coding sequence, we are tuning translation kinetics. And the 3' UTR mainly controls stability. The poly A tail, we choose its length and how we encode, or add. On top of all these sequence designs, we have safety constraints in uridine content, proper capping, and that avoiding double-strand RNA. So, and finally, manufacturing also imposes physical limits. That is, T7 initiation requirements, and you cannot avoid 3' variability. So, therapeutic mRNA engineering is not just one optimization. It is a coordinated set of regional decision, design decisions.